Welcome back to IGCSE Biology. In video 1, we learned what enzymes are, how they work, and the key terms active site, complementary shape, and enzyme substrate complex. In this video, we go deeper. How does temperature affect enzyme activity? How does pH affect it? And what exactly happens at the molecular level? Here is your exam question for this video. Read it carefully. Explain why increasing temperature above the optimum reduces enzymes activity. By the end of this video, you will answer this perfectly. Let's go. Let's start with temperature. Every enzyme has an optimum temperature. This is the temperature at which it works fastest, at its peak activity. For most human enzymes, this optimum temperature is around 37 degrees Celsius, which is our normal body temperature, and it makes sense, right? Now look at these three zones I will be discussing on this slide. Below the optimum. Now below the optimum temperature, the temperature is low. This means low kinetic energy. Molecules are moving slowly. The substrate rarely gets a chance to collide with the active site. So, very few enzyme substrate complexes are forming and the rate of reaction is low. At the optimum, this is the sweet spot. Maximum kinetic energy. Frequent effective collisions between substrate and active site will happen. The most enzyme substrate complexes are forming every second. So the rate of reaction is at its highest stage. Situation above the optimum temperature is this. This is the examiner's favorite part. Now, too much kinetic energy is there. Enzyme molecules literally begins to shake apart. The active site changes shape. So when the active site has changed its shape, it means the substrate can no longer adjust or fit into it. We can say that the enzyme is denatured. Remember the keyword denatured, not killed. Now look at this graph. Because graph questions on enzymes come up in every past paper. The x-axis of graph shows temperature. The y-axis shows rate of reaction. At 0 degrees, the enzyme has no activity at all. Molecules have almost no kinetic energy. Nothing is happening. Now observe in the graph that as the temperature is increasing, the rate of reaction is rising because more kinetic energy is there. More kinetic energy means more frequent collisions between substrate and active site. More enzyme substrate complexes forming every second. At the optimum temperature, around 37 degrees, which is the optimum temperature for most human enzymes, the rate is at its highest peak. Now observe what happens above the optimum. The rate does not just slow down, it drops sharply. This is because the enzyme is being denatured. The active site has changed its shape permanently. And see, around 60 degrees, the enzyme has completely stopped working. It has fully denatured now and it is not working. It becomes inactive. You can say denatured. So when an examiner asks you to describe this graph, use these exact phrases. Rate increases as temperature increases up to the optimum. Then rate decreases sharply above the optimum as the enzyme is denatured. Now look at the second factor, which is pH. pH is a scale that measures how acidic or alkaline something is. 1 is most acidic, 7 is neutral, 14 is most alkaline. Just like temperature, every enzyme has an optimum pH. This is the pH at which it works fastest. For most enzymes, this is around pH 7, which is neutral, but not all. Now look at if the pH goes far, like it will become far below the optimum pH range, or it will go above the optimum range. In either condition, change in pH alters the shape of the active site of enzyme and substrate can no longer bind to it. So the enzyme stops working. Now look at these examples. Pepsin is the enzyme in your stomach which works at pH 2. Your stomach is highly acidic, so pepsin is specifically designed to work in that acid environment. Salivary amylase, which is present in your mouth, it works at pH 7 in neutral conditions. Most enzymes work around slightly alkaline pH of 7 to 8. Catalase works at pH 7 in all the cells. So, you have to remember these key terms. Optimum pH, the pH at which the enzyme works fastest. Write it down. Now, this is the examiner trap time. 2 mistakes, both cost marks every year. Trap number 1. Student writes, the enzyme stopped working because it got too hot. 0 marks. The correct answer uses specific language. The high temperature caused the active site to change shape. The substrate could no longer bind. The enzyme was denatured. Every word in that sentence is a mark. Now trap number 2. This one surprises students. At low temperature, the enzyme is denatured. Wrong. Denaturation only happens at high temperatures. At low temperatures, the enzyme is simply inactive. Molecules have low kinetic energy, so collisions are rare. But the enzyme is not denatured. If you warm it back up, it starts working again. So this is the critical difference. You must note down that at low temperature means inactive, which is reversible situation. But at high temperature, the enzyme has denatured, which is permanent and irreversible. And the examiner tests this distinction directly. So know it. Now let's answer the question from the beginning of this video. Explain why increasing temperature above the optimum reduces enzyme activity. Build the answer step by step. Each point is a mark. 1. Above the optimum temperature, the kinetic energy of enzyme molecule increases. Point number 2. The enzyme molecule vibrates so much that its shape begins to change. 3. The active site changes shape, so it is no longer complementary to the substrate. 4th point. The substrate cannot bind to the active site. 5th. No enzyme substrate complex can form. 6th. The enzyme has denatured and this change is permanent. Now look at the keywords at the bottom. Kinetic energy, active site, complementary, enzyme substrate complex, and denatured. Every one of these words is in the Cambridge mark scheme. Use them and you will collect every mark. Now let's log the 3 things in mind. Number 1. Every enzyme has an optimum temperature and pH. At the optimum, the rate of reaction is at its highest. Kinetic energy is perfect for maximum collisions between substrate and active site. Number 2. Above the optimum, too much kinetic energy permanently changes the shape of the active site. This substrate cannot fit. the enzyme is denatured. This is irreversible. It cannot be undone. Number 3. This is the trap bitter. Low temperature does not mean denatured. It means inactive. Low kinetic energy means few collisions. But warm the enzyme up and it will start working again. That is for video 2. In the next video, we are going straight into past paper questions on enzymes and we will walk through the full mark scheme. If this helped you, please subscribe. See you in the next one.